Welcome everybody to the Wild Mile. We're going into our wonderful winter season. It's not my favorite, but winter is nonetheless very important for things, especially in the Midwest. Normally in the season we have reduced nitrate and phosphate. These are the types of pollutants that come from fertilizer, that come from dog poop, human wastewater, all this other stuff concentrates in the river. In the summertime, we have lower levels of this because all of that algae, all of the phytoplankton, all of the organisms, all of the plants are using this actively in their body and using it to build new body tissue and things like that. When we get into the river, everything kind of drops off, except that we have found in our most recent water chemistry paper, it's a 5-year lifespan of this floating wetland, and we're finding that as the wetland is matured, as the plants are growing in, as these root systems that dangle down underneath the islands are becoming more complex and more dense, we're finding that the amount of nutrient being pulled up by the plants or otherwise deactivated by all the algae, microbes, what we call periphyton attached to the roots, higher concentrations are being pulled out over time. It's not just the plants that are doing this uptake, which is what most people associate with, but it's also these deep, dense root structures. The root structures are creating little zones where maybe the environment is a little bit different, so different kind of microbes can form, and those different microbes interact with chemicals in a different way. So it's all part of this virtuous process. The updates of the data, anywhere from 5 to 7% removal, are the older data sets. As we've added the last couple of years, that's anywhere between 10 and 25%. And that includes both in the plant growing season and in the plant dormant season. We're not getting this enormous drop off in nutrient uptake just because the plants aren't growing. There's all these other kind of complex chemical interactions that are going on in that root zone that would normally be associated with soil microbes, but here we're getting them all in our periphyte and our algae, our phytoplankton, and our zooplankton, and other types of microbial communities. So we, since 2018, with the Peterson Lab at Illinois State University, have been doing a couple different things, but most reliably, we've been using this vertical water sampler. So basically all this does is you're going to have these kind of suction cups pull out on the sides, and this thing is basically going to sit at a certain level of the river and just snap shut at a point where we deem fit. So we can lower this down so that it's just at the surface level, and we can drop this weight that will close both of these shut at the same time, and get a sample of water at the surface level. Or we can have it go up to 10, 20, however deep the river is, get it at a different point there. So we can kind of understand very, very low by the bottom, is it the same as by the surface? And so as the water passes through our root systems from upstream to downstream, we're able to capture this uptake and find, again, that about 10 or 20% of nitrate or phosphate is removed as this water moves upstream to downstream. As we expand this study going forward, we'll probably add more points both vertically, but then also horizontally, to see if what's happening next to our islands is also happening in the main channel, or if it's just restricted to the area around our islands. And if that's the case, how big of islands do you need to potentially affect this whole water body and make everything passing through cleaner rather than stuff that just touches the islands themselves?